as small as an ant, as large as a man, as tall as a tree. In things so dissimilar is found the unit from which all living things are formed. The cell, a great complex of highly organized and specific activity containing all the information and organization needed for its continued existence. A biological factory producing molecules put together in 1,000 patterns, each with its own role. Among the most important of these molecules are the proteins, fundamental to the dynamic equilibrium which is life. Their study and inquiry into genesis and function. Carbon, hydrogen, nitrogen, sulfur, and oxygen. From these atoms, the synthesis of proteins takes place. Myoglobin, one of thousands, perhaps hundreds of thousands of proteins, all formed from molecular chains called polypeptides, made in turn from only 20 different amino acids. Underlying complexity, always simplicity. Valine, one of the 20. Glycine, a comparatively simple structure. Another amino acid, phenylalanine. Each amino acid consists of a backbone and a side chain. The side chain of phenylalanine is an arrangement of carbon and hydrogen. Remove it, and the backbone is left. This backbone has two functional groups, the amino group and the carboxyl group. The backbone is always the same. It is the side chain which varies and gives the amino acid its individual character. Phenylalanine and valine, two amino acids with different characters. Cysteine with a side chain containing an atom of sulfur. Arginine. Tryptophan. This variation in structure is critical to their role. The first step in forming the polypeptide chain from which proteins are made is for the amino acids to join together by the elimination of a molecule of water. From the carboxyl group of one amino acid, two atoms, one of hydrogen and one of oxygen, are eliminated. From the amino group of the other, an atom of hydrogen. Thus, a bond called a peptide bond is formed between two amino acids. As amino acids join together in this way, a chain is formed. The whole nature and function of the protein is determined by the sequence and number of amino acids in the chain. To study a protein, it must first be isolated and with the least possible disturbance of its natural form. Highly sophisticated techniques are demanded for what is a delicate and difficult task. Before a protein can be understood, every amino acid must not only be identified, but located and counted. The introduction of paper chromatography was a major step forward. Earlier attempts to use the classical methods of organic chemistry could isolate only some of the amino acids. With paper chromatography, after hydrolysis or breakdown of the protein, all the amino acids could be separated and identified. Paper chromatography could identify, but it could not measure accurately enough for detailed analysis. Other techniques were required to make both qualitative and quantitative measurement. Chromatography on a column of ion exchange resin is one such method. By this technique, provided the molecular weight of the protein is known, it is possible to calculate the number of molecules of each amino acid it contains. Ion exchange chromatography can establish which amino acids are present and how much of each. But although we can calculate the number of amino acid molecules, their sequence in the peptide chain has yet to be determined. There may be hundreds of amino acids, a prodigious number of possible permutations. With the aid of specific enzymes, the peptide chain is broken in several places. Then, using chemical labeling techniques, the amino acid sequence in each fragment is separately determined. A vast chemical anagram must be resolved. Its solution, the exact sequence of amino acids originally present in the chain. It may take months, even years, before a point is reached where their complete order can be established. Sequences of amino acids can be synthesized, but it still remains an immensely difficult task to build an extensive peptide chain. With existing chemical techniques, the scientist soon reaches a stage where the difficulties of controlling the processes involved become almost impossible. Yet in nature, gigantic protein molecules of breathtaking complexity are continuously created within the living cell. Here, in the nucleus, is the system of chemical control by which all proteins are synthesized. DNA, deoxyribonucleic acid, from which comes the instructions determining the order of amino acids in the peptide chain. Great molecules twisted and coiled in the nucleus of the cell, containing all the information needed to create and maintain life. How do they function? Essentially, the DNA is made up of a chain of repeating phosphate and sugar groups and is a double-stranded molecule. Attached to the sugar groups are organic bases: adenine, thymine, guanine, and cytosine. These organic bases on the separate strands join together in complementary pairs. Adenine joins only with thymine, and guanine only with cytosine. DNA transmits its orders by means of ribonucleic acid called messenger RNA. This messenger RNA is the complement of one side of a section of the DNA molecule and carries all the directions needed for the manufacture of a protein. Also on the DNA, complementing each sequence of 3 bases, are made smaller molecules of ribonucleic acid called transfer RNA. Both come together at the ribosome outside the nucleus in the cytoplasm of the cell. Here are the pools of amino acids. Messenger RNA comes to the ribosome carrying its built-in message from the DNA. Each transfer RNA is recognized by its specific amino acid. It seeks out its complement on the messenger RNA, and thus establishes the correct position of its amino acid in the peptide chain. Every triplet of bases calls for a specific amino acid which is then joined to its immediate neighbor in correct sequence by the forming of a peptide bond. In this way, the peptide chain is formed. The base sequence in the DNA molecule has determined the sequence of amino acids in the peptide chain. This, in principle, is how the genetic code operates. The number of amino acids in a peptide chain may be limited, but a protein may be composed of several peptide chains. There may be 50, often hundreds, sometimes thousands of amino acids in a single protein. The sequence of amino acids in the peptide chain is called the primary structure of the protein. It is the primary structure of the protein which determines its functional form. The functional form is not a simple arrangement of amino acids as implied by the chemical sequence, but an intricately folded and highly specific configuration which must be known if the function of a protein is to be understood. At this point, the methods and skills of many branches of science and technology are needed. X-ray crystallography provides the next step, making it possible to study the molecular construction of those proteins which can be crystallized. X-rays, which have wavelengths comparable with the distances between atoms, are directed at the crystal and scattered or diffracted. The analysis of myoglobin by this technique was an advance of immense significance in protein research. Many thousands of diffracted waves were recorded and fed into a computer in order to form an image. The image which first revealed the overall three-dimensional form of the functional protein. So the shape of a protein molecule was seen for the first time. The way had been opened for the final step. The examination of the complete atomic structure of myoglobin was now possible. More and more diffraction patterns were analyzed, and the computer fed with programs of great sophistication, until every one of the 2,500 atoms was identified, and the spatial relationship of each individual atom determined. The complete structure of a protein, detailed knowledge essential to an understanding of its function, had at last been established. At times, we seem almost to be approaching the central mystery of life itself. Yet all the work that has been done is no more than the key to future understanding. We have established the structure of a handful of the less complex proteins. Now science is concerned with wider study and new techniques, with the next stage, the way in which proteins interact with one another in the control of the chemical processes of life. We are still at the beginning of a journey, and there are no certain routes.